Hello and welcome to the webinar on innovations that accelerate the HLA typing workflow by NGS. I am Selene van der Poel, and as Education Coordinator, I'm proud to welcome you to the Gendex Training School today by participating in this webinar. We started small but are now a team of about 40 people worldwide who are determined to equip you with the best solutions for sequencing-based HLA typing and chimers and monitoring. And now I would like to introduce my colleague, Loes van der Pas, who is Project Manager, Research and Development. She has been with Gendex for quite a number of years now and has been instrumental in the development of the NGS Go workflow for HLA typing on Illumina platforms. And Loes is excited to share with you the latest developments in the NGS workflow, resulting in a more easy and efficient workflow. Loes?
So my name is Loes van der Pas, and today I will be speaking about some of the recent innovations that we made around NGS Go. And the NGS Go products, they are developed by Gendex, and they consist of a range of reagents specifically for NGS-based typing. I will take you first through the NGS workflow to get from DNA to an HLA typing. And what you start with is the target generation, and this involves the amplification of your DNA of interest. So in our case, these are the HLA loci. We have local-specific primers for 11 class 1 and class 2 loci, but also primers for non-classical loci and HLA-related loci such as HLA E, F, G, MIC-A, MIC-B. And we also started a project where we are working on a typing strategy for care. So the target generation, the amplification of your loci of interest, involves PCR reactions, which usually is taking place overnight. And after that, these amplicons have to be processed in such a way that they can be sequenced on an NGS platform. And this process is called the library preparation. So the reagents for the library preparations are called NGS Go Library X and NGS Go Index. And they contain all the enzymes and the buffers, and also the barcoding primers that are necessary for the library preparation. Now we have reagents for both the Illumina and Ion Torrent platforms, but today I will only speak about the optimizations specifically for the Illumina platform. And the Illumina library prep consists of 3 main steps. So the first step is a fragmentation, which is followed by an adapter ligation, and finally a dual indexing PCR to barcode the samples. Now once the library prep has been completed, the DNA is basically a fully functional library that can be sequenced on an NGS platform. On an Illumina platform, this will first require a clonal amplification, which is automatically followed by a sequencing process. Most people that do NGS-based HLA typing use either the MiSeq or the MiniSeq because the capacity of these platforms is very suitable for amplicon sequencing, which is what we are doing here. And we generally apply a paired-end sequencing strategy, and this takes about 16 to 24 hours depending on the flow cell that you're using. And after the run is completed, the machine will generate FASTQ files that can be imported in the HLA typing software at NGS Engine to come to the actual HLA typing result. So that means that after 3 to 4 days you will have come from a DNA sample to your HLA typing result. And looking at the hands-on time, the step that requires most of the hands-on time is actually the library preparation. So this is why we were looking at ways to reduce the time necessary to perform the library preparation to make the NGS Go workflow simpler and also faster, of course. And in this webinar I want to present 3 optimization steps that we made. So we have a new product for NGS Go Index, a different product format where we provide the indices in a plate. We also have a faster cleaner protocol that I will present, and we now also support fully automated workflows where a robot performs all the steps of the library preparation. Okay, so I will go into a little bit more detail about these optimizations. So if you're familiar with our workflow, you will know that the first step of the library preparation involves a fragmentation. And in the same reaction, the DNA, so your amplicon, will also be end-repaired and the A-tailed after the fragmentation. And the idea behind this is that you will end up with DNA fragments that will have an A-tail. And this A-tail is actually necessary for the next step, where the Illumina-compatible adapter, which has a T over N, will be able to ligate to this DNA fragment. And after the adapter ligation, you will have to perform a cleanup in order to get rid of all the free adapters, and also to perform a size selection to select for DNA fragments of 400 base pairs and larger. The size selection occurs on a big magnet that supports 96 wells plate. So up until this step, nothing has really changed in the library preparation procedure. Now, after the cleanup, you will end with DNA fragments. You will end up with DNA fragments that have these universal adapters to each side. But you will also have to provide barcodes to the sequences in order to be able to do a sample identification. And this barcoding is done by so-called indexing PCR. And for this indexing PCR, we use a panel of primers that can yield to these universal adapter sequences. So we have P5 indexing primers and P7 indexing primers. And together, they form a unique combination that provide a unique barcoding sequence to each sample. So for example, if you want to barcode 24 samples, what you can do is take 4 unique P5 sequences, four unique P5 primers, in combination with 6 unique P7 primers, and 4 times 6 together ensures that you can barcode 24 samples with a unique combination of P5 and P7 indexing primers. So this layout supports 24 samples, but if you want to do more than 24, you can simply expand the number of indices and then you can do, for example, 96 samples. And if you want to achieve a very high throughput, we even have enough indexing primers to support the barcoding of up to 384 samples. So, as I just explained, the indexing PCR basically means that you will have to combine a unique P5 and P7 index primer pair to each sample. And this will actually involve quite a lot of pipetting steps, especially when you have a high throughput and you're looking at a lot of samples, 96 or more than 96. This will mean that you will have to do a lot of pipetting. Now, of course, you can use a multi-channel, but we now also have we have a new product where the indexing primers are already pre-allocated in the wells of a 96 wells plate. So here you can see an image of these plates, and each well contains a pre-allocated index pair. So one P5 and one P7 primer. The primers come in a dried format, and as you can see, we also added a color so that you can easily distinguish the primer palette. And what you have to do is simply add your DNA samples and the hi-fi PCR mix, resuspend, and then the indexing PCR reaction mixture is ready to be put in the thermal cycler. Now, each plate supports the barcoding of 96 samples, and we have 4 different plate layouts, which we numbered plate 1, 2, 3, and 4. And each plate type contains a unique index pair combination layout. So with combining these 4 plates, you're able to combine up to 384 samples in one NGS run. And since we have these different plates available, this also supports easy barcode rotation. So just by switching your plate type with each run, you ensure that you have used different barcodes in that particular NGS run. So the plates are very easy to use. Of course, when you do a manual library preparation, you will save a lot of pipetting steps. But these plates also they integrate very well in an automated workflow. So when you're using a liquid handler, for example. Now we'll get into that in more detail in a moment. So if you have any interest in using these plates, I can tell you that they will be available as from next month. So after the indexing PCR, there's another step, which is a second cleanup, and this is also one of the steps that we have optimized. So the current protocol involves a cleanup that I refer to as a plate-based cleanup. So to summarize, so we have performed an indexing PCR in a 96 Wells plate, and after that you do a cleanup in a 96 Wells plate on this big magnet that supports plates. And after the cleanup, all the libraries will be pulled together in one tube, and this will be your final library. Now, what we have done, we now also provide a second alternative protocol where we inverted the last two steps. So we don't pull the libraries after the cleanup, but we already pull the libraries directly after the indexing PCR. And this is possible because the libraries, they are of course already barcoded at this point because the indexing PCR has already been completed. So what we do is, and I refer to this this as a tube-based cleanup. So we do an indexing PCR in a 96 well plate, and then we immediately pool all the libraries in equal volumes in one tube, and then we also do the cleanup in one tube. So the only requirement of this protocol is that you will need an additional magnet that supports 1.5 milliliter tubes. And the advantage is of course that you can only do one cleanup instead of a cleanup on each individual well. So that will definitely save some time. So the protocol is already available and it can be downloaded from our website. And especially when you're processing a lot of samples, it will be up to 30 minutes faster than the current protocol. We still support the older protocol, so the place-based cleanup. So we leave the choice at you. So if you don't have the second magnet, you can still use the old protocol. Or when you have a liquid handler, for example, that does the library preparation automatically and only supports this 96 well magnet, you can remain using this old protocol. Another advantage of this new protocol is that you also need, you use less consumables, so less beads, less pipetting tips, but you need this extra magnet. And what we're using here in the lab, for example, is the Magnosphere from Promega. And of course, we have ensured that this new protocol has the same performance as the previous protocol. So what we have aimed for is that the library yield is the same. And here you see an example of two libraries, one generated with the plate-based cleanup and the second one with the tube-based cleanup, so the new protocol. And with both methods we get more or less the same library yield. Also looking at the DNA fragments, fragment size is virtually indistinguishable. So fragments range from 500 to 1,000 base pairs. And when you look at the NGS data itself, we look at instance sizes, we don't see any effect. Read depths are very similar, and also the mappability is very much the same. So the performance of both protocols is equal. And of course, the most important part, the typing results are also the same. I'm not showing it here, but I can refer you to a technical application note that is available on our website that goes into more detail on this. So yeah, the new cleanup protocol and the new NGS Go index plates, they are of course together a very nice way to reduce the number of pipetting steps to make the whole library preparation procedure simpler and faster. But if you really want to get rid of any hands-on time, you would have to look at automation of your workflow using a robot. And this is why I want to mention that we have several collaborations going on with these automation partners, such as Beckman Coulter, Hamilton, Deacon, but also others. And with a liquid handler from one of these, it's basically possible to automate the whole NGS library prep from start to end without any human intervention. So here, for example, on this image, I'm showing you the automated workflow that we have implemented in the laboratory of Gendex. It's Biomec 4000. And I will not go into all the details, but what you see, for example, here are the pipette heads, the tips. There's a thermocycler. Over here, there's a magnet. And here you can actually see the position where the NGS Go index plate with the new format is located. So again, this shows that these index plates, they fit really nicely in an automated workflow. You can also see that we're not using all wells simultaneously, so it's not necessary to do all 96 samples in one go. So if you want to use less, you just use a subset of the wells, and you can save the remaining indices for another run. So with this automated workflow, we are now able to do a whole library preparation in 4.5 hours with only a minimal time of 10 minutes for setting up the workflow. So this is really a very limited time that you have to invest to do the library prep. Now, so with that, I would like to conclude this webinar, where I've shown you several ways to reduce the hands-on time. So, one, we have a faster, cleaner protocol that will help the reduction of the library prep. And secondly, we also have the new NGS Go index plates that will make the setting up of the indexing a lot faster. So we have index plates in 4 different layouts available as from next month. And these plates, they also are easy to be integrated in any manual or automated NGS workflows. And for any additional information, such as the protocol or the technical note, I would like to refer to our website where you can find all the details.
Thank you, Luz. And while you are thinking of any questions you have for Luz, please, if you have, please put them in the chat box in your GoToMeeting application. And before we turn to the questions and give you a little bit of time to actually type them in, I will tell you a little bit more on what options are available to you for HLA typing by NGS. As Luz has mentioned in her presentation, you can break down any NGS workflow in 5 steps: target generation, library preparation, clonal amplification, and sequencing. For target generation, we offer NGS Go MBAX for amplification of the HLA gene of interest. The reagents are platform independent. You can make any combination of HLA loci, and HLA loci are also separately available. For library preparation, we use NGS Go, LibRex, and Index. These kits are also suitable to produce libraries using a range of different amplicons, not necessarily generated from HLA. So you can combine different experiments in one NGS run to make it more cost-effective and efficient. Clonal amplification and sequencing are dependent on the platform of choice. For data analysis, NGS Engine offers a platform-independent software package which runs on a regular laptop, so there's no need for any supercomputers or dedicated servers. The complete workflow is CE-IVD marked for the Illumina platforms in Europe, and currently Ion Torrent specific library preparation kits are available as RUO, research use only, but we are working hard on completing the CE-IVD marking of these products. If you want to know more or you have any questions, contact us via email, the website, or give us a call, and we are happy to help. You can also find a listing of local distributors and their contact details on our website, www.gendex.com. You can also follow us on social media, Facebook, Twitter, LinkedIn, and Instagram, where you will get the latest news. The next opportunities to meet us are at our next webinar in October, which will be on the highlights of the ESHI conference, which was last week. If you have not visited the conference, it will be the time to digest all of the highlights. The next hands-on training course on NGS HLA typing will be in March 2018 at the Gendex facility here in Utrecht, the Netherlands. And you can even bring your own samples to test NGS to the limit. And finally, are there any questions? And please put them in the chat box. I see there are some questions here. First question, is this new protocol CE-IVD approved?
Yes, so we have the new cleanup protocol that is now part of the CE-IVD protocol. So on the website you will find two IFUs, one for people that would like the RUO version and one for the people that would like the CE-IVD workflow. So yes.
Good. So yes it is, it's CE-IVD approved. Next question, does the pre-coded indices are not available for 24 sample library format? Are these index plates available for 24 samples?
We don't have a plate setup that are limited to 24 samples. So if you want to do 24, I would recommend to use a plate consisting or supporting 96 samples, but then only use a quarter of that plate. And you can store the remaining of the plate in the freezer, and then when you want to do a second run, you can use the remainder. So with a 96 well plate, you will be able to do 4 runs of 24 samples, if that is what you are looking for.
Indeed. So what we have had now, or the indices are either available in the tubes, which is also a little bit more than 24, but you can always store. So you would have enough indices there for multiple runs.
Mm-hmm.
So another question, what about the ion torrent sequencers?
So yes, in this webinar I only spoke about Illumina, but if you compare the workflow of the ion torrent with the Illumina, then the ion torrent workflow is already quite simple. So there is no indexing PCR, and also the pooling already occurs directly after the ligation because we use barcoded adapters. So there's no need to make a choice. You already do this single tube cleanup step.
So in that sense, the workflow for the ion torrent is already quite simplified, and now even more comparable now to the Illumina workflow, because you do the library pooling just after the adapter ligation. Exactly. And before the first site selection and cleanup step. Great. So, and I think also in terms, if you talk about automation, the ion torrent workflow is already, of course, automated in the sense that you can get the Ion Chef instrument as well. But apart from the Ion Chef, the workflow is, as I said, quite similar to the Illumina workflow, and which is already automated. So it can easily be automated on other liquid handler systems, because it is so similar to the procedure used for the Illumina platforms. And I think with this we will conclude the session. I would like to thank you all for attending this webinar, and have a great day.
Thank you, and see you next time.